Hello everybody, so today we're going to be focusing on another macromolecule, and that is lipids. So lipids are a variety of nonpolar biological molecules that are composed mostly of hydrogen, carbon, and lesser amounts of oxygen. Now, they're not really considered macromolecules because they're not as large as carbohydrates and not as complex, and they're also not polymers of some specific defined monomeric subunit like carbohydrates were defined polymers of monosaccharides, but lipids are not really like that. Lipids contain many nonpolar CH bonds, which makes them insoluble in water, which means that they do not dissolve in water. They are soluble in other non-polar solvents, and they're stored by cells to be used as energy sources, and others can be used as hormones that regulate cell activities and serve as vitamins. Now, lipids contain many different categories, so here's just some of them: fats, phospholipids, steroids, cholesterol, and waxes. So first we're going to look at fats, and a specific type we're going to look at is triglycerides. So a lipid is basically made up of two different types of molecules, a fatty acid and a glycerol molecule. Now in a fat molecule, 1 to 3 fatty acid chains can be joined together to a single glycerol molecule through a dehydration synthesis reaction between the OH functional groups on the glycerol and the carboxyl functional groups on the fatty acids, which we're going to see the structure of. So triglycerides are one of the most well-known fats, right? And they contain 3 fatty acid chains, hence the name triglycerides, which are linked to a glycerol molecule. So here we can see that glycerol molecule, and here we can see one fatty acid chain over here. So this fatty acid chain can vary in length. So in this case, it only contains 4 carbons, but this can have several different amounts of carbons. So the reaction between the glycerol and fatty acid occurs between the hydroxyl of the glycerol and the carboxyl group on the fatty acid. And what happens is that the H is removed from the hydroxyl and the OH is removed from the carboxyl, which forms one water molecule, and that allows for this fatty acid to join on to this glycerol over here at this site. And so that's why several different glycerols can join on, and in the case of triglycerides, 3 of them are going to join on, and it's going to form something that looks like this. So here the O from the glycerol is going to bond onto the C of the fatty acid, so right over here, and it's going to form these 3 fatty acid chains. Now, something important to remember is that a fat molecule can have identical or different types of fatty acid chains that are going to be linked together to the glycerol. And so there's going to be 3 different types of fats. So the first type is going to be a saturated fat. So if the hydrocarbon chain of the fatty acid binds the maximum number of hydrogen atoms, and that means that all of the carbons in this case are going to be linked to each other with single bonds, the fatty acid is said to be saturated because it's saturated with hydrogen. So for example, fats that are obtained from animals like butter and lard, they contain only saturated fatty acids. And saturated fats are usually solid at room temperature. The reason being is because of their linear structure, they can tightly pack together. The second type of fat is unsaturated fats, and unsaturated fats are going to have at least one double bond. The fatty acid is unsaturated when there's at least one double bond. So here there's one double bond. And that makes the carbon chain the carbon chain has the potential to actually form more bonds with hydrogen. So the presence of this double bond will create this kink or bend in the molecule. And that's what makes unsaturated fats liquid at room temperature, because the bends and the kinks prevent the tight packing. And so examples of unsaturated fats would be fats that are derived from plants like olive oil that contain more unsaturated fats. Polyunsaturated fats, the last type, we don't have a picture here, but they're basically just multiple double bonds that are in the structure of the fatty acid. And the presence of these multiple double bonds obviously creates lots of kinks in the molecule, and that allows for it to bend even more. So triglycerides function widely as stored energy, gram for gram, yielding more than 2 to 3 times more energy than carbohydrates, and that's why they're an excellent source of energy in our diet. They also provide cushioning to our organs like our kidneys and insulation in animals. So a process we're going to look at now is hydrogenation. And hydrogenation in simple terms is basically just chemically altered fats. And the process of this is basically taking an unsaturated fat and turning it into a saturated fat. Now, how is that done? Well, the synthetic conversion is done by adding hydrogen to make the unsaturated fat saturated. So before we look at the examples, I'm just going to show you the diagram. So here we have an unsaturated fat with the double bond. So the addition of hydrogen allows for this double bond to be broken, and that place or those bonds are replaced with hydrogens. So instead of there being a double bond, this carbon will now bond this hydrogen, and this carbon will bond this hydrogen, let's say, and it's going to form a saturated fatty acid. And so these examples of hydrogenation can be seen in peanut butter, margarine, and this is done to actually prevent the lipids from separating into the oils. But obviously there are going to be bad effects of this hydrogenation process, such as arteriosclerosis, which is going to cause plaque buildup in your arteries that harden over time, constricting blood flow to certain parts of your body. This can cause a burst, leading to a heart attack, and could lead to other types of cardiovascular disease. So next we're going to look at phospholipids. So in the most common phospholipids, as in triglycerides, glycerol is going to form the backbone of a phospholipid. But in this case, only 2 of the binding sites are going to be occupied by a fatty acid, and the third site is going to link to a charged phosphate group, and it's which is often bound to another polar or charged unit. Now, the phosphate group has a negative charge, and the additional polar or charged units could be sugars, amines, and other groups that are going to attach. So here's the basic structure of the phospholipid. Again, just like the triglyceride, except the glycerol is going to be and 2 of its sites are taken up by the hydrophobic tails, and then 1 of the sites is going to be taken by that phosphate group and that other additional polar or charged unit. So molecules actually have both of these properties of hydrophobic and hydrophilic regions. They're called amphipathic or amphipathic molecules. The head is hydrophilic and the tails are hydrophobic, which means that they repel water, and so that makes them nonpolar. So phospholipids make up the lipid bilayer of the cell membranes, which are a very important structural feature of cells. So the hydrophilic end is going to face outwards toward the extracellular and intracellular fluid, and the hydrophobic fatty acid tails are going to face inward toward each other. Another example would be in the case of my cells. And my cells are basically going to have these hydrophilic heads facing outwards, and they're going to encapsulate some sort of nonpolar molecule that needs to be transported through the intracellular, extracellular environment. And so the nonpolar tails are just going to kind of hold on to that and transport it around the cell. The next type of lipid we're going to look at are steroids, and steroids are lipids with structures that are based on the framework of 4 fused carbon rings and a small acyl tail or a carbon chain. And so they're hydrophobic, which makes them insoluble and nonpolar, and they vary in the functional groups that distinguish the one steroid from another. So sex hormones like testosterone, estrogens, and progesterones are all steroids that control development of sexual traits and sex cells that are specific to males and females. And so here is an example of one of the steroids. In this case, this is testosterone, and you could see those 4 fused carbon rings. The next type of lipid is cholesterol. Cholesterol is a very important precursor for all other steroids, and it's also essential for the cell membrane. So here we can see that cell membrane made up of the fatty acids, and so cholesterol is going to regulate the fluidity of the cell membrane. So when it's too cold, the cell membrane would tend to constrict and become very rigid. So the cholesterol is kind of going to space them out. And when it becomes too hot, right, we don't want the membrane to become too fluid. So the cholesterol is going to prevent that extreme fluidity as well. So the cholesterol is just maintaining the normal cell membrane from not being too fluid or too rigid. So high levels of cholesterol can lead to cardiovascular disease, and there's two types of cholesterol we're going to look at. We are going to look at LDL, which is low density lipoprotein, and that's the bad cholesterol. And then HDL is the high density lipoprotein, which is the good cholesterol. So you can actually know the ratio of the two different types of cholesterol to prevent cardiovascular disease. The last lipid that we're going to look at are waxes, which are made up of long fatty acid chains that are linked together to alcohols or carbon rings. They're hydrophobic and extremely nonpolar, and they're actually soft solids that over a wide range of temperatures, and this is going to prevent water loss and dehydration, for example, in the cutin of many different leaves and plants. So this provides protective waterproof coating for stems, leaves, and fruits, as well as on the skin and feathers, and also the shiny exoskeleton of insects, right? So again, preventing water loss and dehydration.